Hey everybody, welcome back to yet another journal club. At one point I kept track of how many journal clubs we did. I don't know how many. It's been 6+ years. What is it? That's more than 10, less than 1,000, something like that, right? Within an order of magnitude. Uh, just as a reminder, everybody, we are fast approaching our Ending Age-Related Disease Conference, which is our 6th annual one. So I've been doing these conferences about as long as I've been doing these journal clubs, just about. And it's going to be here in New York City live. We're not going to let little things like pandemics and COVID get in the way. We're bringing everybody back. That's everybody wants to do it live, so here we are. It's going to be at the, I think, Capitol building, Lower Manhattan, close to where I work, Cooper Union, so Lower East Side of Manhattan. Wonderful place, great venue. There's going to be a lot of phenomenal speakers. So August 10th and 11th. If you haven't booked tickets, shame on you. Jump online, get some tickets, invite your friends, bring everybody over. I'll be there. I hope to be there, barring any unforeseen circumstances or emergencies, but I'm planning to be there. As will a number of other Lifespan.io personnel, including Keith. So if you'd like to see Keith live and in person, he'll be there. With that out of the way, and if you want to take a look at the program, do check out our website. It's all there. I think, yeah.
Yeah, and if you want to get 20% off as well, boy, have we got an offer for you. You can use the code Keep On Thinking, all one word. It's a tie-in with our channel Life Noggin, which is our animated science edutainment channel on YouTube, which has got about 3.5 million followers. It's pretty famous, but yeah, if you use that, you can get 20% off the regular price. Keep on thinking, which is the tagline. And that's about all I've got to say. Hopefully you can join us, but without any further ado, it's back to you, Oliver, for the sports.
Awesome. Okay, so it reminds me of the slogan from the 70s, keep on trucking. So keep on thinking is the new one, I guess. All right. Alrighty. Let's see if I can share my screen. Oh, there we go. Found the paper. Way too many tabs open, as per usual. So this paper is a Nature Biotech paper. People have been asking about it. Steve, you've been asking about it. So here we are. It's a new technology. Well, it builds on previous technologies, right? So it's nothing ever kind of comes out fully formed out of, what is it, the forehead of Hera, or some one of the other goddesses. Everything builds on other work, and this is sort of a kind of a Back to the Future kind of thing, because it uses these proteins called tails, transcription activator like effectors, which these proteins were identified, I believe, in a plant bacterium, a bacterium that infects plants. It is a bacterium, I think that's correct, and they bind to DNA. So before CRISPR-Cas9 came about, these tails and their fusion protein derivative talins, transcription activator like effector nucleases, were being developed as a programmable endonuclease to cut DNA. And then all of a sudden CRISPR-Cas9 came, and everybody started focusing on CRISPR-Cas9 for the reasons I'll get into in a few moments. But this takes advantage of the tail technology to modify it to be a tool to edit mitochondrial DNA, and specifically human mitochondrial DNA. So why are they using these tails instead of CRISPR-Cas9? And what's the deal with human mitochondrial DNA? Well, most of us here know that mitochondria have their own little DNA. They have plasmids, and I think the copy number varies in human mitochondria. I think it's like somewhere between 5 to 10 or maybe more copies per mitochondrium. Can't remember how many tens of thousands of bases it is, like 16 kb, I think. So not too big. So most of the mitochondrial proteins are encoded in nuclear DNA, but a substantial amount, I think it's 13, are located in the plasmids that are found in mitochondria. And mitochondria, other than being the powerhouses of the cell, they evolved. It's been theorized with a lot of evidence that, an endosymbiotic process happened long ago, and now you have these organelles, which are, you got hundreds of them in each cell. They all got their own genomes, and these genomes, are prone to errors and problems. And there's a number of human hereditary, conditions that affect mitochondrial DNA. And the mitochondrial DNA, unlike nuclear DNA, it's in order to access it, you have to get through two lipid bilayers, and you have to go through a transporter or a series of these pore proteins that selectively take up proteins, and in some cases RNAs and other molecules. So if you want to get into this heart of the mitochondria, you gotta go through some barriers. You gotta, the molecules have to go through several portals to get in there, and that makes the CRISPR-Cas9 system a little bit difficult because there's two types of molecular flavors there, right? It's RNA and protein, and so there's multiple transporters that have to be used to import the editing stuff into the mitochondria. And then even if you do that, if you just put in CRISPR and Cas9, you're limited to sort of just cutting the DNA unless you have your repair template, right? So you got to kind of throw in a lot of stuff there to make changes. So also conveniently, a lot of mitochondrial DNA mutations happen to be point mutations, which means you don't really need to edit out huge chunks of DNA, but you can selectively edit, change A to a G, for example, using this process called deamination, which the paper goes into. So you could selectively, so a lot of, don't know the number, I can't remember the number that the authors mentioned in the paper, but it's a substantial proportion, like more than 50% of these hereditary conditions are point mutations. So what these authors did is they developed this, or refined this system to essentially get a base editing system into the mitochondria kind of very expeditiously. So we'll take a little look at that. So strand selective base editing of human mitochondrial DNA using something they call mito-BEs. So mito base editors, I believe that's the term. And they have a couple of variants that they develop in this paper. I think mito-ABEs, mito-CBEs. A's are basically converting adenines to guanines, and C's converting cytosines, cytosines to thymines. I think it's cytosines to thymines. Take a look at the paper. It's been a while since I read this. Hours since I read this. So the lab is not a lab that we typically cover. I believe it's University of Peking in China, Peking University Genome Editing Research Center specifically, and it's the Yi et al. Paper. So I'm going to stop sharing this for a moment, and I'm going to give a little bit of background here on, I put together a slide presentation, lecture mode. All right. So what are these tools, and just kind of a little bit of refresher here. So CRISPR-Cas9, obviously, we've been talking about it a lot. The seminal papers that took this technology that was found in Nature, Bacterial Acquired Immune System, and developed it, popped out about 2012. Papers, from the Doudna lab, so Jennifer Doudna and Emmanuel Carpentier, they received Nobel Prizes, well deserved. So it's been like 10 plus years now, and it's really exploded. And, fundamentally the reason why it's exploded is because it's got two parts, and the part that is really crucial that makes this programmable is
Oliver, can you put this into a presentation mode so it'll be
Oh, yeah, sure. Sorry about that. Let's go from beginning. Boom. Is that good?
That's good.
All right. Yeah, it's not that many slides, so most of them are actually figures from the paper, so we can just kind of scroll through this. So the Cas9 is the part that does the cutting, which here you see this blob, right? HNH and RUVC are the endonucleases. And notice that there's two of these domains. So one cuts one strand, one cuts the other strand, depending on how they're positioned by this guide RNA. And you basically modify the guide RNA to basically have this system go essentially wherever you want, given certain limitations that have been overcome over the years, right? There's this PAM site and so on and so forth, but there's a whole kind of, constellation of these different Cas9 family members that could be utilized. So what the first system did was essentially make a double-stranded break. It cut each of these endonucleases, cut one strand. Once you do that, the cell recognizes this as a kind of a severe damage. So if you were to do this in mitochondria and break the DNA, it would be recognized as a pretty bad damage, and probably the mitochondria would, the DNA would get degraded. They would undergo autophagy. So you'd get rid of that mitochondria. So it's a pretty severe, treatment if you want to treat something that has a mitochondrial mutation. Now, if you have mutations that only are affecting a subset of mitochondria, these heteroplasmic mutations, so not every mitochondria is affected, then you could wipe out the bad mitochondria. But if you have mutations that affect every mitochondria, well, you clearly don't want to wipe out all your mitochondria because then you die. So that leaves kind of a more finessed way of trying to make alterations, which is not a double-stranded break, but having only one endonuclease that cuts at one position and basically just nicking one strand. So if you cut one strand on a double-stranded piece of DNA, that's called a nick. And the enzymatic activity is referred to as a nickase, right? So you won't go under do homology-directed repair or non-homology end joining, as I mentioned before, to plug this gap or switch over to apoptosis and death. But the cell readily repairs these little nicks. There's a whole slew of enzymes that do this. So part of this system involves the application of an enzyme called a nickase. And the reason they require the use of a nickase is that having a nicked piece of DNA in conjunction with another enzyme in proximity targets the surrounding nucleotides to certain chemical modifications and changes, and that's where the deaminase part comes from. So, CRISPR-Cas9 has also been modified into nickase functions by essentially trivially just basically mutating one of the endonuclease domains by mutating in this case, a histidine to an alanine, and basically knocking out the function, and now you just have one endonuclease. So there's CRISPR-Cas9s that actually function as nickases, along with all sorts of other permutations of these enzymes. The other enzyme is a deaminase, which we'll get into in a moment. And what deamination is essentially the removal of an amine, which is found on nucleotides. So here's an adenosine. If this NH2 group is removed, you get it converted to an OH, which gets converted, I believe, into a ketone and the O double bond. And this base is called an inosine or inosine. And the way it base pairs, it's very similar. Well, there's 2 hydrogen bonds instead of 3, but it behaves like a guanosine. It's treated as a G by the cells for evolutionary reasons. So you get this conversion of an adenine to an inosine. And over here is the bonding base pairing you would see with a guanine and a cytosine, and a similar kind of, fairly stable base pairing can happen with a cytosine as well with an inosine, which would not happen with an adenosine because adenosine normally pairs with a thymine, right? So AT. So if you change the adenosine to an inosine, now you have an inosine and a thymine. And that's recognized as a problem by the cell, and the cell then needs to convert one of the nucleotides. It needs to repair it, and on one of the strands, it tends to repair that back into a G. So you end up tricking the system into putting in a guanine where there was an adenine. So it's called an A to G deamination. And you can get a similar effect with cytosines, as we see later. Cytosine can be deaminated into uracils, which are found in RNA. So any questions about this biochemistry? Sorts of other weird stuff.
Yeah. I might have missed something here, but so this will swap the letter on the side that you nicked, right? How does the letter get swapped on the opposite side, the pair?
It doesn't. It tends to prefer one of the strands over the other, according to the authors.
So when this is being read and transcribed into RNA, it just picks one side and uses that one, and the other side is just
not
non-matching.
RNA,
that's okay.
Not RNA, but when it gets replicated. So right now, so this is not being converted into messenger RNA. This is still hard coded in the DNA. So I'm just going to skip to this. So this is their proposed mechanism of action. You have a nick happen here. Here's your TAAT. You have this transient single-stranded DNA, so this little strand pops out here. You have a deaminase which converts these into these A's into inosines, both of them, but only one of them, the one that's on the non-nick strand, and I'm not really familiar exactly why the enzymatic repair pathway prefers to do it this way and ignores this strand. But this strand gets converted back into an A. What it was originally, the one on the non-nick strand becomes the G, which then in subsequent replications, the other strand picks up a C because that's the normal position. So it's only yeah.
So when you do this, you actually have a mismatch.
Yes.
Like a mismatch pair until the next replication, and then when it replicates, it picks one of the two.
Yes. I believe so. Yes. So it has to undergo. My understanding is it has to undergo one round of repair and replication for this to stick.
So does that mean that 50% of the time where you successfully get this far, it will revert back, and 50% of the time on replication? Because the replication is going to pick one of the two strands, right? Or is it well defined which strand it's going to pick for replication?
Good question. I don't know. I haven't gone down that deep into the repair pathway what's on this one.
Okay.
So go back here.
So sorry, still on that. So this, my understanding, and this could just be my lack of expertise in this area or knowledge. My understanding, though, was that they needed to match in order to properly bind together. Does that mean you have basically your double strand is kind of disjointed where they don't match until
the inosine and the cytosine are not really disjointed per se. It's sort of a symmetrical fit. So if you were to have like an A and a G, then it would be disjointed.
Okay, so they still do fit. The two strands still do fit together. Yeah. They still lock together. It'll still zip properly.
Yes.
Okay.
Yes,
interesting.
Yeah. So interesting. I think inosine is I think it's a intermediate in. So it is. It's not like a complete. It's not like a synthetic, unnatural nucleotide. It's found, I believe, in some tRNAs where there's a process called wobble base pairing where some of the tRNAs have like. So inosine can base pair with cytosine, but it can also base pair with like two other nucleotides as well. So it gives certain tRNAs a flexibility for certain codons, and I believe it's also used. It's an intermediate in this biosynthesis of adenosine. So it does. So you have inosines naturally occurring. So yeah,
makes me think of like a wild card in card games. Where
yes.
Whatever you want.
Actually, that's very apropos. Inosine is sort of like is a wild card. I think there's other nucleotide variants that can act as this kind of wild card function. So that's actually a good description. It's the wild card. So how is this wild card introduced? This picture here is moving away from CRISPR-Cas9. This is basically a space-filling model of a chimeric protein, right? A chimera is the ancient Greek, mythological beast of different pastiche of different parts put together, and that's what these proteins are. They're chimeric. And this space-filling model kind of shows you three-dimensionally how this stuff kind of fits together on a double helix, which will become important when they look at which strand gets affected. Because you can see here that it's not two-dimensional. It's three-dimensional, and how you position it, positions these endonucleases in certain positions. This FOC1 happens to be this endonuclease that people have used a lot and still do because it's kind of a non-specific cutter, but these two halves will cause a double-stranded break, unlike a nick. So this is just one variant of this tail-end technology where you have each of these subunits binds to, I can't remember how many nucleotides, it's either 2 or 3 nucleotides, but you basically can make a modular tail-end. This is the programmable part. So this part here, the blue on either side, acts like the guide RNA in CRISPR-Cas9. So this is what gives the specificity and brings all of your other stuff to the location where it needs to go. And this is what made things difficult for scientists, because I think the AI systems now are going to really speed this up, because traditionally designing proteins and modifying them so they can go to different, basically making a unique protein, it's not, it was sort of a, an art and science. You can stick these domains together and say, well, this one binds to an AT, this one binds to a GC, this one binds to a CC, so we can program it and make this thing. But sometimes these don't fold properly if they're next to, or the linkers, which are the connecting nucleotides, are the wrong size. So there's a lot of trial and error to basically get this right, unlike a guide RNA where you basically just synthesize 20 nucleotides and stick it onto a constant region of another of the same guide RNA, and it works like most of the time or all of the time, right? You don't have to really take into consideration too much the three-dimensionality of how all of these amino acids interact. So that's why CRISPR-Cas9 really took off, because it's expensive and time-consuming to reprogram, if you will, these TALENs, these transcription activator-like effector nucleases into a different configuration that goes someplace else. But now, we've seen a paper published a couple of years ago where people have predicted to a very high degree of accuracy three-dimensional, crystal structures using software. So this might make a reversion back to the tails, something worth considering if we can now very predictably redesign new tail sequences to target things somewhere else fairly easily and take a lot of the trial and error out of this. Yes, Michael?
If you solve the problem you described of designing these things, are they otherwise strictly better than the homing mechanisms that are used in CRISPR-Cas9? Or are there some other disadvantage?
Well, they're better in this case because in order to get into mitochondria, it has to go, if you have one protein or two proteins, or if it's a protein, this stuff goes through this transporter complex, which is called, I think it's kind of, the acronyms are TIM and TOM. TOM stands for translocator of mitochondria, I believe, and I can't remember. I think translocator of inner membrane. So there are these protein pore complexes that basically will target these proteins through it. And in order to get CRISPR-Cas9, there's an additional transporter, I think, that was discovered that can transport certain RNAs that have a certain sequence, a special RNA tag attached to them. So I know people are developing CRISPR-Cas9 also to target mitochondria, but it might just be less efficient as far as transportation into that organelle is concerned.
Would it make sense to use these tails in just traditional normal cells rather than not mitochondria, but normal cells as well? Or are they specific for mitochondrial editing?
No. This is used in nuclear editing as well.
Okay, and are they better than CRISPR? Again, if we ignore the difficulty in engineering them, is the difficulty in engineering them the only reason that we choose the CRISPR mechanism? So
it's not my area of expertise. I'm going to say, based on what little I've read about them, it should be just as good as CRISPR. The only disadvantage is that it's just a pain and hassle of designing a completely new protein every time you want it to go someplace else. So the programmable part wasn't so programmable as CRISPR, and also you can and the RNAs, and also it's cheaper too. The RNAs are smaller. You only have 20 nucleotides versus a lot more amino acids that you have to confer specificity to a certain location. So it just really took off. People could synthesize, hundreds of different RNAs and target multiplex sites all over the place, within one afternoon, versus probably taking years with the tails if you wanted to multiplex that many locations.
Gotcha.
Yeah. So here are these chimeric monsters. So in the paper, what they utilize is, when you're making these chimeric proteins, evolution has kind of given us a leg up here, in that when you look at proteins, they tend to be, when they twist and contort and form intermolecular interactions to stabilize their three-dimensional shape. It looks kind of like a glob, but if you look into the glob, there's subglobs, and those subglobs are called domains that are connected with little disordered regions of amino acids. And you can take proteins that have a certain conserved function and splice them onto another protein, as well if you don't interrupt the domain structure, and basically come up with this modular new protein that is one single protein but has several different functions depending on where it is. So in order to get a mitochondria, you need this mitochondrial targeting sequence. So everything in a eukaryotic cell kind of needs a zip code to get to a particular destination, whether it's a nuclear localization signal, or you need to get into the lysosomes, or you need to get secreted out of the cell. You need specialized sequences of amino acids to do that. Then you need, of course, your tail region. So this system here that they developed is a two-component part that gives higher specificity. You have a tail region 1 and a tail region 2, which flank the area that's going to get modified. They have figures that I'm not going to go into, but they're in the supplemental, where they actually make versions of these chimeric proteins that are just one protein, where the nickase and deaminase are on one tail. And the advantage of that is that it's smaller, so you can fit it into adenoviruses or other viral delivery systems, which here's kind of another disadvantage. Like if you were to use this full system here, I think it might be getting a little too big to fit into certain viruses. But the authors say that in some cases you can get away with having the nickase and deaminase on one, and in not for all sites, but for some of the sites they tested, it works just as well. So that's something to keep in mind. So one of the domains is a nickase, as I mentioned, which does the nicking, leaves a single strand, and the other is a deaminase. And the reason they need a nickase is that this A to G deamination really happens way more preferentially if there's a nicked single strand than if you don't have a nicked single strand. So it happens, but at a very low level. So that nickase really accelerates the process. It allows the deaminase to basically latch onto the strand and do what it needs to do, or both strands, the single strand that's kind of lifted off and the other single strand region that's underneath it. So that is basically the platform that they developed. So without much further ado, we can take a look at the paper. So they looked at several different mitochondrial locations, and really most of this paper is it reminds me of the Jennifer Doudna Emmanuel Carpentier paper from 2012, which I went over in class with students like 4 times probably over the years, every single figure. It's one of these papers where it's boring but necessary, right? If you're gonna invent a tool to characterize all the limitations of the tool as much as possible. So basically, move things around at one nucleotide at a time to see where it cuts precisely. Just to characterize it and to figure out all the limitations of it, to know specifically which nucleotide or nucleotides get affected, which don't, kind of so on and so forth. So they kind of really do an extensive job here. I think they use several different human cell lines later. They start with these HEC293 T cells, these human embryonic kidney cells. I don't know why they picked them specifically for this, but they're a common cell line that's used in a lot of studies because of ease of, it's human, it's easily propagated, so on and so forth. But it's a cell line. I think it's a cancer cell line, so you need to obviously test it on a number of other cell lines to make sure it's going to behave. So this is their system here. So these mtRNR2, this is all mtRNR2, but they look at several different loci. This is mitochondrial DNA location. So this is a particular spot on the mitochondrial DNA. I don't know why they picked those spots per se, except that one of the nickases recognizes a sequence, and it's within these sites. And they look at other sites as well. So this TAD8E V106W block, this is your deaminase. They mentioned, I think it's a modified deaminase. So valine gets changed at position 106. It's a modified engineered E. coli, I believe that's where it was derived from, deaminase. These little boxes are supposed to be your tails, and this gray box is probably a mitochondrial localization signal, and this blue box is your deaminase. So here you have two halves that's just a deaminase on both sides. And editing efficiency is a change of one of the nucleotides to another, and it's pretty low, like 0.26 to 0.28%. They mentioned in the paper that it's just barely above like the error of their sequencing runs. So it's like really low, right, this percentage. So if you look at that percentage, only like 0.28% of the mitochondrial loci had a change at either this A or this T or this A. So when they say AT, it refers to either the top strand or the bottom strand. We noticed that it goes up. Yes, Micah. Question.
For are they trying to, in these particular graphics? What nucleotide are they trying to edit? I'm guessing that A5 and A8, are they also trying to edit T6, or is that a side effect in A11?
Well, I think they don't know personally, right? I mean, it's as they say. We shall.
Oh, this is kind of a. This is just a throw things at the wall and see what sticks sort of experiment.
Well, they know that the recognition. So this mute H is the nickase, bacterial nickase, and it recognizes this GATC sequence. It's a palindromic sequence. So GATC in this direction, GATC in that direction. So this nickase needs this sequence, and it will nick, I believe, at I think between either this location or that location, depending on how the mute H is positioned. Exactly which A is going to be changed into a G, essentially. There's a lot of it's variable. It depends on where exactly the mute H and this deaminase are positioned, how close to this location. So that's something that, as they say, needs to be empirically determined. So meaning that they sequence this whole area around here to see, well, what's the preferential nucleotide that actually gets deaminated. So that's part of the characterization. So that's a part that they couldn't predict. Okay. So they couldn't put that into a computer, and the computer says, yes, A5 is going to primarily do this all the time, and is this? So these will change depending on, and that's what they do in the rest of the paper. They move these around depending on where this tail binds. If they move it a few more bases away or closer, then the strand that gets preferentially mutated changes, and these bar graphs also shift around. So it's very dependent on how precisely, or it is down to the nucleotide level, how closely you are to the location where you need to change. Yeah. So when they add the mute H, this goes up substantially, and they mentioned in the paper that at A5 it goes up to like 77% editing efficiency at this location. It's A8, and then there's a T here and an A, which this T is on the other strand. So there are other spots which also get changed to varying degrees. So I don't know if that's too much of an issue. I guess if you're doing base editing, obviously you want specifically one, but at this A5 position, it clearly goes up, orders of magnitude up to 77%. And this nickase activity is essential because if they use a mutated form of mute H, which is this D70A, that's just, again, amino acid jargon, aspartic acid at position 70 getting converted to alanine, it knocks out the enzymatic function. Everything binds, but there's no nicking, and hence you have like very little or no changes, even though you have one of these deaminases here. It seems like it even goes lower than having two of them jammed into this location. So the nickase function is basically essential to get this deaminase to work properly. Editing purity. So looking at these positions, I believe this just means that your A's and T's get converted to G's and C's and not to something else like a CG or maybe back to an AT. So these are T6 is a little bit variable, just a tiny bit, but it seems that the deamination, essentially it's acting as a deaminase primarily. There's no other type of mutation that's being put in there. And this is the model that they had posited, because they mention here that when you have this NIC, this transient single-stranded DNA, you get these inosines put in by the deaminase domain, and the one that is this inosine that's in the non-NIC strand is the one that preferentially sticks, one that remains as a G, and the inosine that was initially put in on the NIC strand converts back into A. I don't know the details exactly why that's the case, rather than vice versa. Somebody here is an expert on nicases and the repair of deamination. By all means, jump in, but I think it'd be a very kind of technical biochemical discussion involving DNA topology and how the enzyme binds, and so on and so forth. And here is the positioning effect. So here are these tails. These boxes are where the sequences that tails recognize. And you can see that they shifted these inwards and outwards, one nucleotide at a time. And you could see this blue is editing efficiency at these positions. This, I believe, is the one that's primarily affected, the AT. But you could see that if you shift them outwards, you get no changes at this location, but you get now a higher editing efficiency at this AT that's outside of this, the recognition site of the mute H. So there's actually 2 recognition sites that are required for this to work. Kind of like the PAM site is also required for Cas9. You need to, and for this particular nicase, it has a sequence that it recognizes, which has to be GATC, or GATC backwards, so palindromic sequence, along with the sequences that the tails recognize to bring this whole complex over. So if this tail brought this mute H to someplace else, like a TTAC, I don't think anything would happen. The mute H wouldn't nick it, and as we saw before, if there's no nicking, you don't have any deamination happening. So it's very this kind of reminds me years and years ago, back in the 90s when I was in an undergraduate. People would do these experiments where they would just like very painstakingly, empirically, trial and error, look at fusion proteins that they had cleaved away bit by bit, nibbled away at a protein to see where certain functions were. And then you can sort of narrow in whether a certain domain, a region is necessary for a protein to have a certain activity. Not a perfect way to do things, but that's the way things were done. Or we still are done to some extent. Yeah, Micah.
Should we be interested at all in the non-zero editing that occurred kind of 10 base pairs after?
Yeah, like you mean like these areas here?
Yeah, there's that line of editing that seems to have occurred. And
yeah.
It seems to be stable across the movements.
Yeah, I'm not sure. I mean, there is a certain, a little bit of messiness here, right? Because you've got this. Whoops. I've got a touchpad here. Sorry. It's just. Yeah. So you've got these bases being edited a little bit outside. You could sort of So there's like a 10% editing efficiency way out here, even though you have a more than 50% editing efficiency here. The authors don't seem to mention that too much. Could be a problem. Obviously, if you change, nucleotide at the wrong spot, could always be a big problem or not a big problem, depending on how important that spot is.
For these tests, were there multiple runs at each location, or is this just one sample per location?
No. I think they did at least three runs, or three cell lines. Yeah, or three groups of cells that were treated.
Okay, which mean, and each group of cells presumably has many mitochondria that were all edited. So hopefully hundreds or thousands of actual.
Yes. Yeah, so if you have each cell, let's say, has on average probably 1,000 mitochondria. Each mitochondria has 5 copies, so let's say 50,000 per cell. It's roughly ballpark, maybe 100,000, and that's one cell, and then you've got 1 million cells in a plate.
So if I understand this correctly, the idea here with this sort of experiment would be that you can identify, so if we target the tail here and here, we only see editing where we want. We don't see any editing anywhere else.
Yes.
There's that one spot's got a really bright blue and the same horizontal line on the y-axis. has nothing else anywhere in it. So we would choose that presumably, right?
Yes. So if we skip through all the way to figure 5, this figure, which I'm not going to spend too much time on, they do like mitochondrial sequence analysis, and also nuclear sequence analysis, because in an earlier version of these base editors, some of these deaminases were able to interact with certain nuclear proteins, so you can get off-site deamination. This is a different type of deaminase that they're doing, and they showed that that wasn't the case. But essentially, they're looking at, so this is position, base pair, frequency of change, and this is like 0%. So they do a sequencing run across the entire mitochondrial genome. I don't know exactly how many they looked at. N equals 3 independent biological replicates. This is ABE, mutH, non-targeting. This is something else that they do later. This is another deaminase, the cytosine to uracil deaminase. Only when they have these variants that are targeting to these locations, when they do these sequencing runs, they say they don't pick up anything statistically, any statistically higher sequence changes outside of the region. I don't know exactly how they bin the region, what size it is. This might be, as you mentioned, Micah, it's like you have about 20 or 30 nucleotides on either side where you can have some A's get changed. So, and this is 15,000. So at least far enough away, you don't get these changes happening in the rest of the genome. So anyway. Yeah. Okay. Let me look at this. Yeah, we did. Oops. Apologies here. Yes. Okay. So, so the proximity of how you position. Obviously, how you position these tails will alter the frequency of of whether a certain A is deaminated eventually to a G, and also which strand. And I think it's probably depends on. So I mentioned that it's primarily the non-nicked strand, which gets, so it says deamination A remains preferentially in the non-nicked strand after DNA repair and replication. This is just their schematic showing that. And here they have a tail mutH, which is 0 to 4 bases away, very close proximity to the GATC site where the nick happens. And then this one is 5 to 9 bases away, a little further away, where the nick happens on the opposite strand. And just to highlight, the three-dimensionality of this is like, well, why is the nick happening on that strand if you move it away? What's the deal? It's probably because if you look at DNA as a double helix, right? Every twist and turn of the double helix is about 10.5 nucleotides, right? So if you're here's a nick, but if you move your whole system this way, it'll rotate to the other side of the double helix, and the nick has probably been happening on this strand. So that was my trying to topologically get my head around as how the enzyme probably sees it, right? So you're as you move things around, you probably swivel around to the other side. So this is all context dependent. So which strand gets preferentially deaminated, and it depends on how you shift the location back and forth. Any questions about that? Okay. Yeah, this is just a figure I got, and I just slapped the GATC right over it, so a little bit messy there. So that's the mutH sequence. Okay, so you have this mutH sequence, this GATC recognition sequence for mutH, and I think it only lies in about 7% or so of the mitochondrial genome. So if you want to mutate the rest of the locations, then you're out of luck with mutH. So what the authors do now is they try to mutate mutH and look at other nickases that have other sequence recognition sites to basically build an expansion pack for these enzymes, right? So here's your wild type. They look at a bunch of, so this structure here is the sequences that they're looking at, the the R groups, aspartic acid, lysine, so on and so forth, that are in close proximity or touching this GATC, right? So this is sort of the active area, and they look at various mutations at various amino acids, and when you deactivate a certain interaction, usually they convert it into an alanine, which is a small hydrophobic nucleic acid. Amino acid. And you can see that these two here, so phenylalanine to alanine, E, what's E? I forgot what E was. K is lysine. Just got their single letter chart in front of them. R is arginine. Anyway. So E91A, F94A position, retains essentially the editing efficiency. These basically get rid of it. So they made this mutant. I don't know if for any other reason other than they still recognized. I don't know why they, guess they decided to go with a double mutant, right? And it seemed to have enhanced editing efficiency at this site, this GATC site. So they wanted to see whether or not it could recognize other sites. I don't know what other reasoning they used to predict that it might bind to other sites as well, but they went ahead and tested it, and the answer is that it did recognize fairly well other sites. So mtND5, so another site in mitochondria. So I think they were also testing several variants of these tails, positioning this double mutant, which they now call mutH asterisk, right? So this is your double mutant mutH. And here's your right one tail. So they tried it on the right, tail and the left tail. And they, basically analyzing this mutant to see where the editing efficiency would lie. And this is a GATA site instead of a GATC. And lo and behold, also fairly high editing efficiency at these new sites. So when they look at here, so it's dependent on where these tails are. So if they have it on the top strand, it doesn't work. It might work depending on how they position it, right? So this is within pretty close. So maybe if they moved outwards, it would change. But so let's see. Do they have a control here? I don't think so. Just wondering if they. Well, we know that I think they tested earlier that the mutH wild type wouldn't recognize anything outside of GATC. So now you have recognition at this GATA site, particularly at this T location, with some smaller degrees of editing efficiency outside of that. They also, this same mutant mutH asterisk recognized at GATG sites at MTCO2 location with a lot less off-site targeting. So that's that's interesting. That's good. Oops, sorry, this is really touch sensitive. And GATT, right? So they've kind of expanded the repertoire of this mutH asterisk. So they mention here and they summarize that if we look at just your mutH that's non-mutated, you get a 6% coverage of the mitochondrial genome, and that goes up to 71% of coverage if you have this mutH that's been modified with these two amino acids. Average 2, what is that? Numbers 0, 2, 4, and 6 indicate the frequency of mutaging recognition sequences within a 40 base pair region. Okay, so you get much more coverage. So their expansion pack worked to that degree. So later they decide to further expand this. So they look at a whole bunch of other nicases, a whole bunch of other variants from different species. A lot of strange names here. Some of them look like they're engineered variants. So nicases that basically have very weak or no recognition sequence restrictions for mito, base editing. And there's this one particular one that they use that this NTBSPD6I parentheses C. Who named this? It probably refers to some species of bacteria. And yeah, so anyway. So they go on. So this nickase here that they're referring to at mtND1 and these other locations, I believe they're looking specific. So these are all the nickases, and they're getting a much more efficacious activity from this NTBSP-D6I C nickase that's now being shuttled to these locations. And they basically now look at it, in the context of a whole bunch of other sites to also further characterize, which of the A's or which of the T's, if, depending on which strand and how close it is primarily being affected. So further characterization of these mito base editors. So further expands everything here. So we'll get a whole bunch of other sites. So on and on and on. Next, what they do is you've got A to G transitions. So deamination from an A to an inosine, then conversion to a guanosine. But now they're looking at a different, so they've been sticking with the same deaminase. Now they switch to a different one called RAPOBC1 to deaminate cytosines into uracils, which then the cytosine gets converted to thymine. And basically it's kind of a similar but different process, right? You have a CGs normally are interacting. You have a UG, which you now have a misspare intermediate, so it's a little bit different with the in deamination here. You have a mismatch, which is not very conducive to proper base pairing. And then you can, since the uracil sort of acts like a T, you can convert a G to an A and now have a proper base pairing. In the cell, what usually happens is you have excision repair. So a uracil doesn't normally exist in double-stranded DNA. So there's these enzymes, these glycosylases, which basically literally chop out the nucleotide at the riboses, at the deoxyriboses, and then we'll stick back a C. So you get it, so once a U pops up, if this deamination happens somehow, so either spontaneously or through some side reaction, these excision repair enzymes will get it back. So if you really want this to stick in this direction, you have to add in something else, which is this uracil deoxyglycosylase inhibitor. So excuse me. So there's a protein, I think it's like they mentioned, it's 80 amino acids, which blocks the function of these UDGs, these repair enzymes. So it doesn't cleave out the uracil that you just deaminated. It basically sticks there, so that way your deamination goes all the way to a C to a T transition. So a lot of stuff happening. So in this back to figure 4, that's what this 2X UGI is. It's yet another chimeric protein that has mitochondrial targeting sequence, a tail specific to that location, your C to T deaminase, your RAPOBC1 in this case, and 2 of these UGI stretches of amino acids, because I guess 2 is better than 1, it works better, right? It's kind of a joke, but not really. You can sometimes get better activity by increasing the localized concentration of certain enzymes to a limit, right? Before things just get too unwieldy. So probably a series of experiments were done earlier to show that 2 of these stretches is better than 1. So you now have basically the same characterization, but now you have a conversion at the C location rather than the A location or the T location, right? So, and depending on the strand that you're looking at, it could also be, the G strand and vice versa. So I just boxed the positions where this, where these peaks happen. So that is that. This one here, so they look at also this DDCPE. I don't really want to go too much into this. I think this is like a split deaminase that they were studying to use as a control that does not prefer one strand or the other. So I think it was used in earlier. I mentioned earlier that this one has an issue where it can interact with certain nuclear proteins. I'm not 100% sure why they use this as a control here. I think it's used as a control. It was more to point out that their deaminases, you can tailor them to specify one strand or the other, versus some of the other ones that were these split deaminases that don't have a preference for one strand or the other. So you tend to have both strands get changed, or one or the other in equal amounts. Figure 5 we went into, which they they wanted to make sure that the rest of the genome was untouched, so they do sequence analysis. They mentioned that they have average coverage of, what's the average coverage here? I think they sequenced it like, that's the mean, so over 1,000 times, sequencing runs. So if you're into sequencing, there's a certain minimal amount of times you want to sequence things over and over again to make sure you get full coverage. They also sequenced the human genome. They also looked at to make sure that mitochondrial DNA levels didn't change when they compared it to, I think, a beta-actin, may have been a plasmid control, that when they added these, you didn't see any aberrant genomic changes. I'm not sure why you would expect that, but it seemed like that was normal. So they do mention in the paper that you can always do even deeper sequencing runs to make sure there's no other off-site targeting events, but at least within their capability and sequencing, they didn't see anything outside of that window that we highlighted and looked at that seemed to suggest that deamination was happening elsewhere. And last but not least is the application of this technology. So this kind of bumps it up to the next level. If there's one thing that has come out positively from COVID, is kind of the acceptance and the rapid progression of utilizing variants of RNA, either circular RNA in this case here, or other forms of RNA, to transiently deliver some therapeutic. In the world's case, a vaccine, or several series of vaccines. In this case, the delivery of these tail fusion proteins. So tail mut-H, tail TAD-AE4, EV106W, and these are transfected into cells, and they were looking at cells from an individual with this is a hereditary illness where it leads to, I think, progressive blindness, LHON. What does that stand for? Leber hereditary optic neuropathy. Okay, so LHON was the first disease to be associated with mitochondrial DNA point mutations and is therefore maternally inherited. So there you go, something I learned, that LHON was the first disease to be associated with mitochondrial DNA point mutations. Leber hereditary optic neuropathy. I doubt you'll ever see that question on trivia night in some bar, but if you do, there it is. Okay, so these point mutations are, so one of the reasons that they were looking at this technology is, I mentioned that these point mutations are common in many diseases, particularly LHON. And the types of mutations that can be fixed happen to be, at these locations where you need to undergo a deamination of an A to a G, right? So they have a disease model here. At this point, it's kind of trivial. What you're really, testing is how effective is this delivery of your circular RNA, and then what's the editing efficiency, right? I mean, we already know that if you have a GATC, than this system, and it gets into the mitochondria, it's going to do its job, right? So they look at a number of, so these models, I believe they probably induced these mutations in these cell lines. So I don't think they got these from, let's see, cells derived from individuals, HON. Well, this is HEC293 cells, so I'm wondering which one of these came from an individual with, there's H1299, MCF7, 7, and RPE1. Let's see, what do we have here? So which of these cells actually came from an individual? These are cell lines, definitely the HEC293 cell. So you can definitely put in a mutation to simulate LHON. Let's see. One of these probably, or maybe it's in a later figure. But anyway, so they characterize the editing efficiency, so you could see here that, yeah, it's 77% at HEC293, but it falls off depending on the cell line. So I'm not really certain as to what, what the difference is between these different cell lines. Let's see, where do they derive? So, okay, so here we go. So now we're, what are we looking at here? So tail tad AE06. So here's your, so that's your deaminase. Where's the TBSP? So what is that? Your cytosine deaminase? No, that's your, sorry. So many of these acronyms here. I think that's your, that is your non-specific nickase. And GM10742, I believe this is probably primary cells with an individual LHON disease. So let's see, H. Yep, LHON disease cells, genomic DNA collected 3 days post-trip. So they look at the editing efficiency of these cells, and they notice that, let's see, E is, which one is E? I think one of this, so these are your LHON cells, so 20% was corrected at this location, so this affects the histidine, so you're converting it from a histidine to an arginine, which makes it normal. So, 20% seems low, but again, in some of these mitochondrial illnesses, you don't need to have 100%, especially if it's not a dominant mutation. If it's a mutation that's, well, I can't really, well, yeah, use a dominant mutation, I guess, referring to mitochondrial. So if it's a mutation that doesn't screw up anything else, if it's just simply, a problem with, the respiratory, electron transport chain, then any improvement is an improvement, right? And you'll basically regain efficacy. I think in this case here, they were seeing if they can use the system to screw up respiratory chains. So methionine is your start codon for particular enzymes, and convert it to a C. It's no longer a start codon. So you can also use your system to obviously break certain proteins in the mitochondria and mimic certain respiratory deficits as well. So basically, what they do in the rest of this figure is to characterize that. Well, when they either broke the system or repaired it, they the functioning of the mitochondria improved, and you've got this untreated and non-targeting targeting, and then you've got this curve here, oligomycin. These compounds target various electron transport chain enzymes, and then you get this oxygen consumption rate curve, which I looked up, and it turns out that this is the type of assay that's evidently done, for mitochondrial function, to measure oxygen consumption rate, minimal and maximal. You basically block things and release them to measure, how many picomoles per minute, and this is referred to as a seahorse assay. I'm not sure why it's called a seahorse assay. Doesn't look like a seahorse. Usually it's, they give it a cute name.
Seahorse is the company name that makes the analytical equipment.
I see. So it's nothing to do with the shape. So usually, yeah, okay. So it's not like, It's not like the French flag model for proteins that that are released in developing embryo, right? So depending on the concentration. Okay, cool. So they performed this assay from the current slide, and so non-targeting is up here. Targeting, you have a complete sort of a big drop in oxygen consumption rate. So this assay is not really functional. And then for the cells that have come from LHON, you basically this assay is indicating that some functionality is regained in the electron transport chain. How much, I don't know don't really know exactly how to interpret the oxygen rate here, but it seems to be affected by the various compounds that you add. So the electron transport chain is back in operation, albeit still at a low level, but you get relative ATP goes up a bit, but still significant. So you get partial repair of these LHON cells using this novel technology. So with that, they mentioned that with all the stuff that they developed so far, this A to G transition, or C to T, depending on which deaminase they use, they can potentially get up to 87% coverage of the entire mitochondrial genome. And of course, no doubt, more work needs to be done, to further fine-tune the system.
The last picture, I don't think it was 87% of the genome. I think it's 87% of known diseases, right?
87% of known diseases. Oh, I think you're right about that. Yeah. So.
Like we know these.
Yeah.
1,000 diseases exist. We can hit 87% of them with the handful of tools we built.
You're right. Yeah, 87% of known point mutations, and that's right. And these are the percentages of the mutations. So for that lie for each of these diseases, so pretty good. If much better than it was. So if we can, which is, I think,
it seems like the specificity. Seems like the specificity is the biggest problem. Like all the charts had off-target effects kind of all over the place. There's very few that seemed really super targeted, and whereas in contrast to CRISPR, which the latest versions of CRISPR have very, very, very low off-target effects these days.
Yeah.
But hopefully they'll step in the right direction. Hopefully we'll get there.
Yeah. Well, I mean, this is extremely promising. Given how much how many sites they're able to target with the amount of iterations that they've done. I don't want to just keep waving the AI flag, because I don't really know about artificial intelligence systems, but certainly those types of systems can help further fine-tune these types of tails and maybe modify the deaminases as well to confer even tighter specificity. Again, this is going into all mitochondria, so potentially you might be, you'll be hitting mitochondria that have these defects, but you'll probably be hitting mitochondria that don't have defects. Although, again, if your mitochondria are, all homozygous for this mutation, so if this maternal mutation affects all of your mitochondria, and you're fixing 20 or 30% of them, and you might be putting some other off-site target mutations here and there in some of the other mitochondria, you might have a net positive, right? I mean, depending on the severity of the disease, Yeah,
especially if the offsites just result in mitochondrial death. The loose mitochondria, they replicate, you get them back, but you have more good ones now than
exactly. Yeah. So I can see this. I can definitely see this being rolled out for early clinical testing for really severe variants of mitochondrial diseases that are homozygous throughout, that are homoplasmic, right? So all of the plasmids have the same mutation where you really have no other alternative but to do this. So, then the question is the delivery, right? So there might be certain mitochondrial dysfunctions that, again, LHON, it leads to blindness. So certain cells are probably require a much higher oxygen consumption have the ATP demands are much higher, and if they go below a certain level, you might have neuronal death, and other tissues might be a bit more tolerant to oxygen deprivation. So, again, there's some flexibility there when it comes to delivery that you might, not need to target every single cell in the body for this to be efficacious for a particular mitochondrial dysfunction, as long as you can target those those RNAs into a certain subset of cells that are most detrimentally impacted by this type of mitochondrial mutation, then you can certainly substantially improve the quality of life, and the longevity of that individual who's suffering from that disease.
Yeah, I think one of the things that I'm concerned about with mitochondrial editing, I'm very glad to see it advance, but there's a lot of mitochondria in your body, massive amount. And
well, there's
if
50 trillion cells, right? And then right, you multiply 1,000 by 50 trillion. And
yes, you got a very big number.
Yeah.
You got a big number that most people don't even know what the word for is.
Yeah.
And so I'm worried that trying to hit all or a substantial portion of mitochondria is hard. And as you said, there's some situations I can see where you only need to hit all the ones in your eye, for example. Maybe that's sufficient for a particular disease. But I just worry that it's going to be really hard to translate this, just due to the volume. I mean, just the volume alone. Even if you do find a good delivery mechanism, you got to deliver a lot of whatever it is, and your body generally doesn't like a lot of anything being added to it, right? And so you need something that is completely inert in terms of immunogenicity for your delivery vector and everything. And you probably need to spread it out over a very long period of time, and so you need to have no sort of immune memory to it that builds up. I just worry that just translational is going to be even harder than the CRISPR stuff. But again, very good research.
Yeah.
I think the other thing is for mitochondria, you've got things like mitochondrial transplant, because mitochondria do kind of transfer around whatnot pretty reliably, and you basically can just inject someone with healthy mitochondria, and that seems like an easier path. Just get a donor of a source somehow.
I gotta look at those papers. I gotta look at those papers because I know it wasn't that long ago that people had noticed these weird mitochondrial transport mechanisms of one cell transporting mitochondria to another. It's a fairly recent, I think,
there's
discovery, right?
Yeah, there's the way I know of it is some companies are trying to build out this exact thing, like for dealing with mitochondrial diseases and whatnot, or just loss of mitochondria with age, as you kind of get less and less as you get older. The idea is you just inject a person with a bunch of them, and they seem to, as far as people can tell, they seem to just find their ways to where they're needed. And we don't know why, but it seems like the cells that need mitochondria seem to pick them up, and it just magically works. It's one of these things, very early research, but it seems very promising, at least from the initial studies. And so again, just from a translational standpoint, that feels like it might be a better option. But that being said, maybe this type of technology makes the most sense in extracellular therapies where, you need to modify the mitochondria in some way outside of the body and then you put them back in. And then you don't have to worry so much about the mutations because you can filter or something like that, which is, for a lot of cell therapies, that's how they do it, right? You have some super risky, high off-target effect therapy. You do it outside the body, filter out the bad ones, put the good ones back in. Very interesting stuff, though.
Yeah. Yes, I agree with some of the comments. It is a very technical paper. Cell biology, but getting into the biochemical weeds, we could have certainly gotten even deeper into the biochemistry, but we only got so much time to plow through so many papers and review papers, right? And, I don't think we want to start going into nucleophilic attacks and revisiting organic chemistry, but I think this level suffices for now. Certainly I think this gives an appreciation to our viewers that when you do develop a new technology, particularly these types of proteins, there's a lot of work that, we all know there's a lot of work involved, but there's a lot of, I don't want to say mind-numbing, but it's very, repetitive, repetitious, meticulous work that needs to be done to fully characterize these systems. And if you do read the original CRISPR papers, like by Doudna et al. And others, they go in through this type of detail as well, using in vitro assays to characterize, which section of the guide RNA is important, down to the nucleotide level, which sites, are crucial, because, it is an engineered biological, system. So you need to really fully characterize it. So we're kind of getting, going away from the basic science and, more into the engineering, with these types of modified proteins.
And getting something to just an engineering, just a simple matter of engineering, is a very big step, because engineering is very rote. Like, it's just like, you know what you need to do now, just go search for all these different, do all the tests that they've done, just do more of them, find more of those mutate or whatever things that work in different areas, test, iterate. It's very linear. Like, you can just have a project manager that lays out, the next 10 years, and you can have a plan, you can just follow it, and you get to your destination. Whereas the basic research is much more, let's try some stuff. We don't know what's going to work. And so it's nice to get to the engineering stage, I think.
Yeah, eventually there'll be like a shrine to in the lab, and you'll just walk up to it and say, "Oh, mighty AI, based on everything that you know on deaminases, can you develop a engineered deaminase that targets position number 235, the A to G, transition, without affecting anything else?" And it'll give you the sequence.
Yes, that's still like at least 3 years out, Oliver. Come on, don't be crazy.
Yeah. Maybe even 5.
Maybe even 5 that day.
Yeah, right. Yeah. Okay. Cool. So right now people use a lot of techniques to develop these things. Trial and error is a big component. Looking at the structures, right, and basically using your own personal knowledge base to see which amino acids to shift around, along with software that can help you predict what a change would happen. But yeah, these software tools, as they become more and more powerful, I anticipate to see a much more rapid explosion of the development of very functional and very precise chimeric proteins, much more so than in previous decades, where it's been really kind of a mind-numbing kind of boutique approach to, and still is, as in this paper, to designing all of these variants. With that, this is a new technology, a new tool, and no doubt it's going to develop, and for all the reasons I mentioned before, we'll probably see a much more expanded use of other programmable endonucleases, not just CRISPR and Cas9 variants. So with that, I have nothing else to add. So thank you, everybody, for joining me.
Thank you, Oliver. That was great.
Thanks. So next paper, I don't know what the next paper is, but I have had my eye on the latest paper from the Sinclair lab, where they looked at a cocktail of chemicals that sort of mimics Yamanaka factor activation of modification of the epigenome. So I'm gonna take a look at that paper and if there's nothing else like earth-shattering that comes out between now and later, that might be the paper that we do next. Because yeah,
I did take a look at it. It was interesting. I think it was not nearly as interesting as the media made it out to be, unfortunately. Like, it was a good iterative step. Someone took some research that existed before, iterated on a little bit, some studies, useful stuff. But I think then the media went and took that and we basically solved epigenetic reprogramming in humans. And it was very disappointing when you actually go and read it.
I see. Well, since you took a look at it and read it, without going too far down the rabbit hole here, how did they manage to achieve a really tight specificity or Yamanaka-like factor, activity by using compounds that I would assume would have a lot of other off-target sort of activation happening?
I don't know the answer to that. I think it's been a bit since I read it, a week or two. But if I remember correctly, they just basically tried a bunch of stuff and looked all over, found some things that looked promising, used them. They seemed to work. It didn't seem like it was a very intentional, engineered, we sat down and designed this very specific cocktail. It was more like, hey, we found a thing that seems to work.
I'm laughing because
that's the impression I got.
I'm laughing because the original Yamanaka paper from 2006 and I think the follow-up in 2007, I read that one when it first came out, or a year or two after it came out, which obviously gave Yamanaka and I don't know if any of his collaborators, but definitely Yamanaka, a Nobel Prize, well deserved. But that paper was also extremely, I don't know if you read that one, Micah, but that was also extremely iterative. Like they literally did a literature search and said, okay, what are all the compounds that everybody uses to basically induce pluripotency? And there's several hundred, and they're like, okay, let's just start paring them down until they basically came up with OSKM. And that was and then characterized and said, okay, does OSKM actually convert differentiated cells into a pluripotent state, and now let's do all the standard tests that are done to determine pluripotency, and there you have it. So it was right. It was just very brute force approach.
Yeah, I mean, I do think that there's value in an approach. I think more broadly, it works. I think more broadly, I'm not particularly optimistic about small molecule reprogramming, because once you get into humans, small molecules always have side effects. This is very different from proteins and genes, which seem to be much more targeted. Small molecules just go everywhere; they hit all sorts of places and things. And so in this paper, they just do ex vivo or in vitro, with senescent cells. Even they don't even use just healthy old cells; they use intentionally senescent cells, and so I'm very skeptical that this will translate at all to anything in vivo in humans. Again, I think it's good research. It's an iterative step, useful knowledge. I'm not optimistic that it's going to, go far for many reasons.
Gotcha. Okay, well then perhaps we'll look at another paper. There's always a whole bunch of other papers to look at. We have no shortage of that. That's a good thing, actually.
Just to be clear, I'm not against going over it in Journal Club at all. I just want, for anyone listening, I think I just feel the media makes it out to be more than it is. Good paper, but not some of the articles I saw were we are going to cure aging soon now because of this brilliant discovery that just happened. They were really blowing it up. It was the most amazing thing ever. And I personally think some of the work out of Sinclair Lab that was previously done, the eye regeneration one, was far more groundbreaking than this one. Got less publicity.
Well, it depends on how you spin things, right? I could add 10 years to most people's lives, and everybody tunes in, and I'm like, stop smoking.
Diet and exercise.
Yeah, exactly.
Yes. The answer that no one likes.
Nobody wants to hear that, even though words like, boo, where's the magic pill?
That's right.
Okay, well.
Yeah, I also briefly looked at the paper. And while it's interesting for further exploration, one of the things is that they just measure epigenetic age and they show it's decreased, but what does that actually mean? There is no evidence that we currently have that if you decrease someone's epigenetic age, that translates into an improvement in lifespan. That's something quite different. And they don't show that, for example, mitochondrial function will improve, or any other assays that measure cellular health. They haven't done anything like that, let alone that they have gone to a whole animal and shown that they had a lifespan extension from this cocktail. They haven't done that either. So in that sense, it's a very preliminary paper. And yeah, you will definitely need a lot more research before you can get excited about it, I think.
Excellent. Okay, well, as Steve has mentioned, we are now bleeding into next month's journal club, which and I have to take off actually and do some checking up on my own here. But I will thank you everybody for participating. We will pick yet another cool and awesome and different paper. I don't think we're going to look at another base editing paper for next month. I predict it'll probably be something different, but definitely within the lines of the scope of longevity and aging, either a tool that can help ameliorate it or learn something about it from a fundamental biology perspective, or another kind of fundamental biology paper on aging, as we did with the whale paper and DNA repair. So all of that is really cool, and I will see everybody next month after our Ending Age-Related Disease Conference. I believe the Journal Club will be sometime at the end of August, and our conference will be August 10th and 11th. So I will be here in New York City, and I hope to see
See you in New York. See you in New York.
All right, awesome. Okay, everybody, take care.
Thanks a lot.
Bye bye.
Bye bye.
Gotta run. See ya.